Okay, so now that we have done some protein synthesis, DNA replication I think is going to be a walk in the park. So let's start by reviewing the structure of the DNA itself. The DNA is in a double helix. It has two backbones. Both of those backbones are made out of sequences of phosphates and sugars. Phosphate group, sugar group, phosphate group, sugar group. Those are bound to each other via phosphodiester bonds. That bond is not being represented in this image. We are demonstrating base pairs independently of each other in this particular image, which is not too convenient. Our nitrogenous groups are always going to correspond specifically to each other. A thymine is always going to bind to an adenine, and a cytosine is always going to correspond to a guanine. Those base pairs are oriented towards the midline of this DNA double helix, and they do have hydrogen bonds along that midline, which is our weakest form of bond. It's not going to be too difficult to break that apart. Now, in order to fully understand the structure of the DNA, you do have to understand the concept of 3' and 5'. The way this sugar backbone, this pentose sugar, is oriented, we do have the ability to actually break some of these bonds and make it linear. And when we make it linear, as a rule, this is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, and this is carbon number 5. When we participate in our sugar phosphate backbone, we're going to have a 5' end. That's the one associated with the phosphate group. And we're going to have a 3' group. So that 3' is right in the middle of this pentose sugar. This 3' and its corresponding hydroxyl right there, or hydroxide, is going to participate in the phosphodiester bond with the next phosphate group. So the next phosphate group goes down here. And in this direction, we have our phosphate group up here that's part of this nucleotide. That phosphate group is going to bind to the next 3'. So when we synthesize our DNA, when we're undergoing synthesis, we're always going to attach our next nucleotide to the 3'. We're always going to go from here to a phosphate to a sugar, and then that 3' is going to go to another phosphate to another sugar. So we're always adding on in this direction. So now that let me go this direction. I'm mirrored, so this direction. There we go. Take a close look at what's happening here. Here's our 5', here's our 3'. We bond to this one at its 5'. Here's our 3', this bonds to another one. We're adding new nucleotides in this direction. If you notice what's going on over here, here's the phosphate, here's the sugar. It's, from our view, upside down. Do you see how it's upside down? And do you know why I'm making air quotes and saying it's upside down? It's not really upside down. This is a long, thin strand. It's like a noodle. It's a piece of spaghetti on your plate. So there's no up or down. But in terms of our orientation, or a more general way to phrase it would be, we're always going to have 5' to 3' in opposite directions. If we're going 5', 3' this way, then we're going 5', 3' this way. So we're going in opposite directions. This one's going 5', 3' down, this one's going 5', 3' up, or vice versa. It's always going to be in opposite directions. And that's what this is trying to convey by putting this pentose upside down to us. So once again, we have our DNA double helix. We unwind a segment of it. We break the hydrogen bonds. And then DNA polymerase comes along and adds on the corresponding nitrogenous group for nucleotide. We're always going to get that base pair that matches up. So once again, if we have a C over here, we have a G over here. And we're going to synthesize from 5' to 3'. So we do have some enzymes acting in this activity. DNA helicase binds to DNA and unzips the two strands. DNA polymerase binds to one of those strands and starts adding on corresponding nucleotides down the chain, one by one, in the 5' to 3' direction. We're always going to go from that 3' carbon to the hydroxyl group. We're going to have that dehydration reaction. That's going to be a phosphate on the other end of that. And that's going to be how we always go. So again, if replication is happening this way over here, then replication is happening this way over here, even if it happens to happen in fragments. Because it's much easier if it's being unzipped in this direction to just go with it. If it's being unzipped in this direction, every time we get a new segment revealed, we're gonna do it segment by segment. Did that make sense? I kind of feel like I should repeat that. So the overall direction of replication is this direction. DNA helicase is unzipping this way, so it's very easy to add on new base pairs in this direction. But as soon as we reveal this next fragment, then we can start synthesizing in this direction on this strand, and then the next segment's going to come in when we unzip down here. So hopefully that made more sense. Once again, 5' to 3'. We're going to add on the next phosphate group here, and then we'll have another sugar, and then another phosphate group, and then another sugar, and then another phosphate group, and then another sugar. So a new nucleotide gets added on at that 3' nuke location from 5' to 3', from 5' to 3', 5 to 3, 5 to 3, 5 to 3, 5 to 3. So that's the 5 end, that's the 3 end. And in the other direction as well. Yeah, this just has kind of the same thing twice, doesn't it? Yep. So again, just another way to illustrate this. Eventually what you're going to end up with is two, hopefully, genetically identical strands. So this is DNA replication. This is the synthesis phase. So we'll start talking about mitosis and meiosis in just a bit. And we do have interphase with G1, and then we have S phase, and then we have G2. This is S phase. This is what's happening in S phase when we are preparing for mitosis, when we're preparing for cell division. That's when this takes place. We unzip it, we make two identical copies, 5' to 3', using DNA polymerase, and we end up with two new strands that should be identical to the parent strand. So here's a good video. Hopefully this link works. Let me know if it does not. Again, seeing this in 3D animation is always more beneficial than just learning it from pictures. As previously mentioned, I think I went too far into this in the last video, and I kind of am going to assume to an extent that it's going to cover it here. A mutation means a replication error. All we did was either dropped out a nucleotide or we replaced it with the wrong nucleotide. Now, and again, the effects could be none, it could be beneficial, or it could be dangerous. Some examples, and this is a nice elaboration on what we covered just a bit ago. So the cell could die as a result from having some non-functional protein. That absolutely happens. It could be a disease state. A really common outcome of mutations is cancer. Now, because this is so dangerous, you do have mechanisms in place to repair the DNA. So we have several mechanisms. You may recall that there are checkpoints in G1 and G2 that check for mutations, and if a mutation is found, apoptosis is initiated. We really don't want things to be highly mutated if we have a healthy functioning organism, right? So we do have a lot of things, and that's just one mechanism. There are also specific enzymes that are looking for copy errors, looking for abnormal base pairs or abnormal locations of things, and sometimes we are able to repair those mutations, put the correct nucleotide back into place, and have a functioning strand of DNA that is just like it was before the mutation. So those mechanisms do exist, but obviously cancer also exists, and also evolution definitely exists. So based on that, we know, well, based on all of these things, they are all related. So mutations are happening, and those enzymes and cellular processes are not always 100% effective at preventing them.